blood, how many jobs does it have? Man, oh man, you know, if you think about it, what is blood? If I were to take blood and analyze it, I would find that, you know, and I took it and I centrifuged it, what I would find is that when I ended up, I would find that about, a little bit, let's just say it comes right up to here, probably a little bit more than half would be a liquid portion of the blood called plasma. And about 45%, so about 55% would be plasma, and about 45% would be cells. And let's not forget some of the functions of these things. Plasma is merely the liquid portion of your blood. And what's in plasma? Well, what's in plasma is all the stuff that this whole unit has covered on the transport system, things like nutrients, wastes, some hormones, some proteins that do different jobs, and of course the cells consist of red cells, which you know have to do with hemoglobin and gas exchange, white cells, which have to do with the immune system and immunology, and a group of cells a group of cell particles I want to tell you about right now, platelets. Because you know, every once in a while we run into something sharp, or we run into something that scrapes us, or we run into something that breaks open our capillaries. In fact, sometimes we even break open our capillaries under our skin. We call that a bruise, when the blood leaks out under our skin. But I don't want to talk about that. I want to talk about what happens when you bleed, and why the majority of people can stop that with a blood clot. So we want to talk about blood clots. Here's the scoop. And it starts with these things called platelets. What is a platelet and what does it do? Well, remember collagen? Collagen is a protein. Over half the proteins in your body are collagen. Okay? It's that long, fibrous protein that holds things together. And when you break tissue, that stimulates the clotting process. It turns out that when you break a capillary, step one, capillary is broken. Collagen, the protein collagen, is exposed. And that starts a cascade of reactions, many of which we don't completely understand yet. Why is it are you saying to yourself, why does everything have to be so complicated? Why can't you just say, form a clot? Why does it have to be 27 steps or however many steps there are? Well, would you want your clotting process to accidentally happen right now? Would you like to form blood clots in the middle of a capillary or artery that, say, feeds your brain? I don't think you would. And so the point is that this whole idea, just like the digestive enzymes being inactivated or deactivated and then activated, and just like blood clotting, you don't want it to happen accidentally, because that can mean the end of your existence. So anybody who has accidental blood clotting is going to die and not have the chance to pass their genes on. Natural selection will rear its head. So that's why. Sorry I diverged, but I heard some of you complaining. All right, so capillary broken, the collagen is exposed. What's going to happen next? Well, we're going to get a collagen-platelet interaction, all right? And here's what's going to happen. Here's the capillary, and the platelets are going to literally attach to the collagen, and they're going to begin to swell, all right? So the platelets swell. So step number two, all right, step number one, we break the capillary open. Step number two, the collagen's exposed. Step number three, we're going to get the platelet, which, as I mentioned, are not cells. They're actual fragments of cells from something called a megakaryocyte, in case you were wondering. Platelets and collagen, and the platelet swells. The platelet is now going to release, and this is where we're going to do a cop-out here because of the enormous complexity of this reaction and the things that are still not understood. What's going to happen next is a series of chemicals are going to interact. What are these chemicals? Well, they're called clotting factors. Where do clotting factors come from? Some of them come from platelets. So the platelets are going to give off clotting factors. Turns out some of them are formed by the tissue. And some of them are already there, perhaps some of them formed in the liver. So some are already in your blood from other organs. So the damaged tissue, the platelets, and the other organs, like the liver, has already formed these clotting factors. And now we get some very interesting things happening. Here's what happens next. There's a chemical in your blood. It's one of the clotting factors called prothrombin. It's a protein that, like so many other proteins, is inactivated until stimulated. And prothrombin is broken down into thrombin. Yes, it is. But that doesn't happen unless the clotting factors are there from the platelets, etc., that I just told you about. So now you clot? Nope. Okay, now you're just going to basically have the platelets stuck in this area, starting this reaction beginning to occur. But now the thrombin is going to do something. And this is very interesting. More blood proteins. There's another blood protein floating around with nothing happening called fibrinogen. And fibrinogen can be converted into a fibrous material, a really fibrous material called fibrin. And guess what you need for that to happen? Thrombin. That's right. Unless you go prothrombin-thrombin, you have nothing to catalyze the reaction of fibrinogen to fibrin, and you're not going to go fibrinogen to fibrin without thrombin. And where are you going to get your prothrombin from? Clotting factors. And you're not going to have clotting factors till you damage yourself. Wow. But that's not all. The fibrin is a sticky material, very much, the platelets got very sticky as soon as they started to swell. They stuck to those collagen fibers. And you're going to get this. The fibrin is going to actually form a meshwork, okay? And the meshwork is going to trap, a sticky meshwork, and it's going to trap red blood cells. They're going to get jammed in there. Look at these red blood cells. In fact, if you don't like this, look at this. This is an electron micrograph. We've enhanced it a little bit by adding colors, because you don't get colors out of electron micrographs. But look at how gorgeous that is. Look what this shows. This shows a couple of things. Number one, it shows the fibrin, right? And the fibrin has trapped the red blood cells. You are looking at a scab. Whoever thought a scab could be so beautiful? That is one gorgeous scab. And so that's a scab. We call it a scab, but now you know what a scab truly is. You see that it's trapped these red blood cells. What I love about this picture is it teaches you something about red blood cells. Look at their shape. Look at their nucleus. You don't see a nucleus, do you? That's a good thing, because red blood cells don't have a nucleus. And so the red blood cells are trapped right in there. Red blood cells are perfectly adapted for carrying oxygen, and that's pretty much it. But they make a great scab, because then they'll get stuck in there, they'll form a dam, and now you stop bleeding. Scab eventually dries up, skin heals, and you're on your way, we hope. And of course, on a serious note, there are genetic diseases where people cannot heal properly, hemophilia. When people have liver problems, as in cirrhosis of the liver, quite often they can't heal properly because their liver functions are depressed. And so this whole idea, once again, structure follows function. The whole idea of the platelets releasing clotting factors, clogging that thing up, the fibrin holding that clog in there, forming a dam so things can't get out. It's an unending theme. The body's constant struggle to maintain a static, dynamic situation.